Now let's learn about cloning. Early first clone became only to 6 cells created the advanced cell technology in 2004. Cloning first human clone practice was created and cloning activated into fire. And this protective cloning cloning is used to produce cloned embryos which are implanted in the human form with the intent to create a fully formed human clone. Therapeutic cloning uses the cloning procedure to produce a clonal embryo, but instead of being implanted in the womb and brought to term, it is used to generate stem cells. The four effective types of cloning are DNA gene cloning, adult DNA cloning, therapeutic cloning, and embryo cloning. Recombinant DNA technology for human viral vector carrying healthy gene, cell with defective gene, vector inserts healthy gene into cell, new gene in the cell along with old gene. Embryo cloning is a medical technique which produces homozygotic identical twins or triplets. It duplicates the process that nature uses to produce twins or triplets. Abnormal cells are removed from the fertilized embryo and encouraged to develop into one or more duplicate embryos. Twins or triplets are thus formed with identical DNA. This is a medical technique which produces homozygotic identical twins or triplets. It duplicates the process that uses nature to produce twins or triplets. Abnormal cells are removed from the fertilized embryo and encouraged to develop into one or more duplicated embryos. Twins or triplets are thus formed with identical DNA. And it has been done for many years in various species of animals. Only very limited experimentation has been done on humans. Proposals for germline engineering combine the use of stem cells and embryo cloning. Then we get stem cells harvested and cultivated, viral vectors carrying new genes, colonies grown from each stem cell test colonies for successful incorporation of new genes, then stem cell with new gene, genetically engineered design and or new thinker, cloning process, same result will be produced. Then comes therapeutic cloning or the biomedical cloning. This is a procedure whose initial steps are identical to adult DNA cloning. However, the stem cells are removed from the pre-embryo with the intent of producing tissue or a whole organ for transplant back into the person who supplied the DNA. The pre-embryo dies in the process. The goal of therapeutic cloning is to produce a healthy copy of a sick person's tissue or organ for transplant. This technique would be vastly superior to relying on organ transplants from other people. The supply would be unlimited, so there would be no waiting. The tissue or organ would have the sick person's original DNA, and the patient would not have to take immunosuppressant drugs for the rest of their life, as now required after transplant. There would be an danger of organ rejection. Human therapeutic cloning, patient somatic cell biopsy, nuclear transfer, nucleated donor oocytes, and embryonic stem cells, pancreatic island cells for insulin production, then hematopoietic cells, cardiomyocytes for heart, neurons, and hepatocytes. Immunologically comparable transplant. Then the patient with the disease, somatic cell types required for patient with disease-free cells recovered from patient, transfer of cell to egg and cell fused by electric current, embryo cultured for 7 days, embryo stem cells recovered, embryonic stem cells grown and cultured, specific cell types derived from embryonic stem cells. Adult stem cloning or reproductive cloning. This technique can be intended to produce a duplicate of an existing animal. It has been used to clone a sheep and other mammals. The DNA from an ovum is removed and replaced with the DNA from the cell removed from an adult animal. From the fertilized ovum, now called the pre-embryo, is implanted in the womb and allowed to develop into a new animal. This may or may not have been tried on humans, but is specifically forbidden by law in many countries. It has the potential for producing a twin of an existing person. But based on previous animal studies, it also has the potential of producing severe genetic defects. For example, later recent evidence of medical ethics consider it to be profoundly immoral practice when done in humans. When cloning donor egg, nucleus removed from the egg, genetic material DNA taken from cell person to be cloned, eggs fused with the DNA, stem cells removed from embryo used to treat Alzheimer's, heart disease, and diabetes. What are the risks of cloning? Reproductive cloning is expensive and highly inefficient. Cloned animals tend to have more compromised immune function and higher rates of infection. Genomes of cloning abnormalities Some changes in the normal activation process called editing chemically marks the DNA from the mother and father, so that only one copy of the DNA with the maternal or paternal gene is turned off. Defects in the genetic imprint of DNA from a single donor cell may lead to some of the developmental abnormalities of cloned embryos. Pre-implantation genetic diagnosis and action (PDS) fertilized egg tested for presence of disease-causing genes. Healthy zygotes implanted on board the fetus. 2. May do the same thing as humans with the gene both the mother and the potential fetus and unacceptable high risk of damage. How many abnormal babies would have to be produced to be modern day contain the rising things consequences of instrumental especially working introduction to this objection of the idea of producing traditional it just raises other problems. Instead of being reduced into products of both parents, it would not be the biological child of both parents and the mother. In psychological experiments in relation, if the clone feels that he or she was just a copy of someone else who has already existed and operated themselves, am I really someone else who was put into a different being? What will be my relationship to one I was cloned from? No one can predict with any degree of assurance that the morality of human reproductive cloning will fruitful and multiply, assuming to be natural, but IVF and cloning not mentioned in the Bible. Problems with cloned animals most suffer premature aging and other genetic problems that are already why cloning humans is ethically unacceptable. Controlling someone else's genetic makeup. Child can reject any aspect of its upbringing, but it could never reject the genes that were chosen for it. Such control by one human is never over another. It is incompatible with the ethical notion of human freedom in the sense that each individual's genetic identity should be immediately unpredictable and unplanned. Human cloning would bring acute risks to abuses to human integrity and exploitation of